started. It is my very great pleasure to introduce Professor Arkhat Abzhanov. He is a professor from the Department of Life Sciences at the Imperial College of London and Natural History Museum. His research group is interested in a variety of topics related to vertebrate head development and the evolutionary development of craniofacial features. His group uses a variety of tools, including morphometric, molecular, cellular, and genetic approaches, in order to understand the precise mechanisms of cranial skeletal cell differentiation and skull and face morphogenesis in amniotes. We're extremely pleased to have him speak in our seminar series, and we're very much looking forward to having him tell us about the growth and form of the animal face. So please take it away.
Thank you, Kathleen. Thank you, Alexandra, for inviting me. It's a big pleasure to be part of this seminar. I'm myself a biologist, so from the introduction, but I'm very keen to work with people coming from other fields, including applied mathematics, applied physics, and biomechanics. So what I will tell you today about is a couple of our projects, from a range of projects we're working on, where we really benefited from this collaboration with applied mathematicians, where there's a real biology problem, actually going back many years, and having a very different angle, a very different look from biology and from mathematics perspective was actually really fruitful and gave us additional insight. And the main problem here is trying to understand diversity, more specifically diversity in the faces that you see on Earth. And the faces, it's my favorite part of the body. It's extremely fascinating. It has very rich evolutionary history. Each face tells you something about the species, to the point that you can actually tell what species you're looking at. It has a lot of clade-specific, species-specific, even individual-specific characteristics. The face can also tell you what this animal is doing for living. So the predators and herbivores, many other different types of animals have very different faces. There is a particular pattern to variation, however. For example, you can see variation in facial length. So here I'm comparing mammals in the top row with squamates. These are lizards and snakes. With aves, so these are birds. And finally, crocodilians in the bottom. And all of these animals are showing a huge amount of diversity. And here I'm focusing on the facial length. Some faces are very short, and some faces are medium length, and some faces extremely long. Every single face is adapted for very different types of foods. Like the face on the top, for example, in the corner, the bat with a very short face, it's a food-eating bat, whereas the anteater on the opposite to it in the right corner is very well adapted for introducing its face into the termite nest and licking out the termites from the inside. So I'll tell you more about some of those stories. But again, the key problem here is how we understand this diversity, how we explain it. My main interest is on the animal face and head. And if you look inside the face and the head, it's all about the cranial skeleton, or what we call the skull. And this is a series of bones and cartilages which protect the brain. And if you look at the structure of the skull and face, there are several parts to it. So this is a crocodile face here, the crocodile head. So there is a part which is behind the eyes. There's a portion which protects the brain, which we call the brain case. There's also a portion in front of the eyes, which we call the face. So that includes all the area around the eyes and everything forward to it. And of course, the area immediately in front of the eyes is what we call the snout. So those are some of the terminology that I'll be using in my talk when I talk about the face. Now, I want to switch gears a bit and talk about how this attempts to model biological diversity, model biological shapes, they actually has quite a bit of history. Over 100 years ago, Darcy Thompson, who was a famous Scottish and British mathematician and biologist, he wrote in this, he basically introduced some really interesting methods in his book called On Growth and Form. So in the middle here, you can see the original edition of this book. On the right, you can see one of the later editions, and it was a very well-received book. You can see here a description that Nature magazine gave on Thompson's book. This book on growth and form, it was substantial and stately, is to the credit of British science, an achievement of its distinguished author to be proud of. It is like one of the Darwin's books, well considered, patiently wrought out, learned and cautious, a disclosure of the scientific spirit. A great book. It's actually a very good read. I highly recommend it. Now, in this book, he talks about how he was modeling shapes of how the basically different biological shapes changed, and he used skulls as many of his examples. In fact, there's this very curious book that I found in the library at St. Andrews University that was pointed out to me by a colleague of mine who works there. This is a book by a famous zoologist, American zoologist called Henry Osborn, who wrote this book called The Age of Mammals in 1910. And this is a book which was gifted to Darcy Thompson. You can see at the front page here, it says here, to Darcy Thompson from his friend Henry Osborn, January 1911. So this is 6 years before the publication of the On Growth and Form. And you can see if you actually look, if you look carefully at the illustration of this book, this is illustrations of several mammalian skulls. If you look really close, you'll see this faint outlines of a geometric grid, which are placed around the skulls, which is fascinating because that's exactly what the approach which he was using very widely in his book, where he was trying to understand within geometric space how the entire skull changed. For example, how the skull of the primitive horse, shown here on the left, this was the most ancestral horse found at the time in the early 20th century, how this evolved into a modern horse skull. And he put the original skull on the square grid, and he showed how it had what parts had to be transformed in which way, scaled in which way to produce a modern horse skull. And he also he applied the same approach to ontogeny, to development. So on the right, sorry, you can see the skull of a newborn chimpanzee. If you put a square grid on it, you can trace which parts of the skull have to change in which way for this skull to turn into the shape of the adult chimpanzee skull, which is shown right below it. And it says he also was curious about what's governing this. What are the mechanisms which explain these transformations, these genetic transformations? And he writes in his book that these transformations of biological shapes constitute a proof that variation has proceeded on definite and ordinary lines, that a comprehensive law of growth has pervaded the whole structure in its integrity. So he's talking here that there must be a mechanism, there must be underlying mechanism that explains this. And of course, now it is development biology that is trying to provide that mechanism. So if you look at the face from development biology perspective, the face is also very fascinating. It has a very different strategy how, for example, the bones and cartilages are produced. This is a chick. So on the left, you can see the outlines of the chicken skeletal parts in the head. In blue are the cartilages, which are inside the skull. In red are the bones, which cover the brain, they cover the face, they cover the jaws. Those are flat bones. And these are produced from a very specialized population of stem cells called neural crest cells. During development, these originate from a neural tube. As it closes early in development, they separate from the neural tube. They cease being neuronal. They become mesenchymal, highly migratory cells. They migrate towards the face. They populate the face, and then they begin to grow outward in this series of buds which fuse with each other, and they will eventually grow into what we recognize as the animal face. So if you look inside, if you do a section through the face, you'll see that again. Unfortunately, I cannot see my own cursor, so I don't even see where I cannot even point at things, unfortunately. But if you look at the panel C, on the left, you can see the early embryo, where before the neural crest cells migrate there, it's essentially the space between the brain and the outside. And then in blue, the cells begin to proliferate. They begin to grow outward, and they form structures such as the beak, such as the lower jaw, other parts of the face. So this is the briefly kind of developmental paradigm that underlies the growth of the face. Now, we'd like to combine this to explain some real life interesting evolutionary questions. And one of the main questions, going back to remember the diversity challenge, is to explain diversity of Darwin's finches. Why Darwin's finches? Because since they were discovered and described by Darwin and his colleagues back in the 19th century, this became the textbook example of many different evolutionary phenomena. For example, adaptive radiation, where from a single ancestor, from a single colonist, which came to Galapagos about 3 to 4 million years ago, many more species evolved, each of which has a distinct bill of particular size and shape that is derived, and it's adapted for a particular purpose. So some of these birds have elongated bills for drinking nectar from flowers. Others have very robust bills for cracking seeds, yet others have pointed bills for catching small insects, and so forth. Every single species here has a distinct diet, which is matched by distinct bill shape. So shown here, some of the examples. Again, you can see the large ground finch, which has this very deep and very broad bill that it uses to crack seeds. There's a cactus finch, which is with a pointed bill, which is it's using to get to the nectar inside the flowers and the pulp inside the fruit. There's a vegetarian finch, which is using this very deep but very narrow bill. It uses them as garden shears to cut young leaves and eat them. Woodpecker finch has a very straight bill, as the name suggests. It basically normally goes around woodpecking for larvae under the bark. If it finds a larvae too deep inside the hole, it will look around. It will make a tool from a twig just the right length, and it will use it to take out the larvae from the hole. So again, every single species has a specialized bill. We know from work on zebra finches, which is our laboratory species of songbirds, which we use from which we can collect embryos, because we can grow this, we can keep these birds in captivity and collect the embryos. We know that if you look at the very early bill on the left, the earliest ones, the bill is this very short potato-shaped structure, which is full of proliferating cells, which are shown here as bright green dots. So there's a lot of proliferation, a lot of cell proliferation happening inside the early beak primordium. As the beak begins to elongate and begins to take shape, what you see is that the cellular proliferation acquires a very particular geometry. So only the distalmost cells continue to proliferate until there's a very small growth zone left at the tip of the bill, and about at late stages of development, by about stage 37, you can see that particular organized growth zone disappears. Zone of high cellular proliferation disappears, and then the shape is now completely is finished. So you have this very particular shaped bill. From that point on, the bill continues to expand, but it continues to expand asymmetrically. That is, it increases in all directions in the same way. So it maintains its original shape and just increases in terms of size. So there's several stages here in the middle embryonic stages are critical for establishing the shape of the bill, this particular shape of the zebrafinch bill. Moving on. Okay, hopefully it's not stuck. There we go. It's just slow. Okay, so if we were to have a two-dimensional sort of sideways perspective on the bill, what we see here is that this is a very early developing beak. There's a very large growth zone shown here in green, and as the growth zone begins to decay in size relative to the rest of the bill, you begin to acquire this conical shape, and at some point that essentially becomes a knot, and that forms the tip of the bill, and you have the overall conical shape structure, which is the envelope essentially of all the shapes of the growth zone, certainly from two-dimensional perspective of the which ever existed inside this developing beak. And this formula, this is referring to our early collaboration with Michael Brenner, the applied mathematician at Harvard, with whom we looked at the kind of two-dimensional aspects of bill, just looking at the curvature, looking at the length and the depth of the bill back at a few years ago. So this idea that the overall, what you see in the adults, the shape of the bill, including its curvature, is actually a reflection of the shapes of the growth zone insides. Definitely made sense when we looked at the growth zones inside for example, these very deep-billed species like the Jaspisa magnirostrus, the ground finch. It has a very deep, very broad growth zone, whereas the sharp-billed finch on the bottom has a differently shaped growth zone. So certainly that fit really well. Now, so this was all about the two-dimensional modeling, and we wanted to come up with a way to model bill shape that would explain both its main characteristics. For example, we found that the bill as it grew, basically, first observation is that if you compare in 2D, if you compare the profiles of the two-dimensional profiles of the beak shapes, they all scaled on top of each other, but they fell into three distinct groups, scaling groups. And and which would require additional shear-based transformation to collapse with each other. And again, I'm not sure why it's not moving forward. Oh, here we go. Again, so this is the that's what we're trying to model. Here is a growing beak. There is a growth zone inside. We calculated the decay rate for this growth zone to understand the kind of developmental dynamics of this. We looked at the overall shape, and the consensus equation shown on the bottom, which tries to take into account the observed variation in the beak shapes. That is, again, beaks scale on top of each other. They also require shear to explain the full diversity of the beaks, and they also scale as they grow because of the expansion of the growth zone. But this was all two-dimensional. So more recently, we set up another collaboration with Dr. Mahadivan's group, also from Harvard, who wanted to. So we wanted to look at three-dimensional shape of the bill. So for that, we scanned the skulls of all the species of Darwin's finches. So we obtained this high-definition three-dimensional CT scans, computer tomography scans, and then we turned these CT scans into meshes. So we specifically kind of removed the beak and we focused the beak, although you'll see that kind of actually the overall skull shape became very important as well in this work. But we just wanted to focus on the beak, and this you can see on the right is the phylogeny. So as I mentioned earlier, every single beak here is distinct. It has distinct curvature, distinct characteristics, and that's what we wanted to understand. We wanted to see how much of the two-dimensional also how much of the two-dimensional modeling was still applicable. And this is the paper which I believe literally just came out today from this collaboration. It's called Geometry and Dynamics of Link Form Function and Evolution of Finch Beaks. So I'll tell you some of the kind of more important findings from this paper over the next few slides. So again, we wanted to take this landmarkless approach because this is a kind of very structure which has a lot of curvature to it. It's very two-dimensional. It doesn't have a lot of defined landmarks, and we took this mesh and we wanted to understand how these beaks compared across all the diversity of Darwin's finches. Here's a kind of three-dimensional distribution of these beaks, looking at key characteristics, including the curvature, including curvature along the length of the beak, but also curvature along the width of the beak, because both curvatures are very important. Some beaks are very curvy, going to the front. Other beaks are actually can be very curvy but very narrow as well. So both curvatures are very important for the eventual adaptation. Now, going back to the developmental model, remember I told you about the growth zone. So as the beak is growing in the embryo, there is this population of cells in the apical part of the beak, in front of the beak, which is proliferating much more rapidly than the rest of the beak. And this model, which we produced in two-dimensional, suggested that the cells should behave very differently when they proliferate inside and outside of the growth zone. Inside the growth zone, the cell division polarity, that is the direction in which the cells divide, when they separate from each other, the two daughter cells separate from each other up to cell division, have to be aligned with the overall axis of growth of the beak, so that the beak continues to grow forward. Once the cells find themselves outside of the growth zone, the behavior should change dramatically. That is, the cell division polarity should basically alter so that they only divide primarily perpendicular to the main axis, so that they expand in all directions isometrically. That is, this would allow for increase in size of the beak without changing its overall shape. So this was, you can see kind of somewhat more advanced 3D modeling based on our observations from growing beaks in zebra finches. And this allowed to produce a model which included cells, which included, and by controlling the cell division polarity, allowed us to produce differently shaped beaks, both in terms of exactly how the cone looks like, extended cone or very deep cone, and also the curvature of the beak, and allows us to produce kind of this model, which was derived from our finches, was in principle turned out to be quite useful to understand other beak shapes in other birds. A little bit more about this later. So another aspect of beak morphology, that of course it does a particular function. In finches, in the Darwin's finches, one of the main functions is to crack a seed. So they put a seed inside the beak and they crack it, and they have to apply a lot of force. And if you look here, there's a little blue dot inside this model, or inside this kind of more abstract model. This is where the seed usually goes. In the finch, that's where the forces have to be applied. So in order to crack a very hard seed, the forces, we know where the muscles attach. We know kind of the upper beak, unlike us, our upper jaw is fixed permanently to the face, but in birds, it actually swivels. This little red triangle shows where the joint is. So the upper beak can actually move relative to the face. So, and they can swing up and down, and they can apply particular forces. And the shape of the beak is designed so that they can apply maximal forces. They maximize the amount of force to that particular point in the beak. And and the beak has to be functional enough to crack the seed, apply a certain number of newtons to crack the seed, but also be tough enough to survive. The beak itself has to survive that operation. So what we found is that we found that particular genetic ratios, such as the depth to length ratio, but also width to depth ratios, turn out to be very, very important. In particular, the Jaspiza magnirostrus, the large ground finch, which has the deepest and the strongest bill, as you can see here, it maximizes its depth to length ratio, and that allows it to produce maximal mechanical advantage, where these forces kind of coming in and the outward and inward balanced forces, they are maximized. So and other finches, they fall along this curve that describes mechanical advantage of the beak. So one thing which is very important here is the angle at which the beak is attached to the this particular angle between this surface here. So I think I'm going to show it better here. So the angle between the beak itself, the main axis of the beak itself, but also this angle of the what's called the jugal bone. So the steeper this angle is, the more force you can produce up here. And this can be achieved, this steep angle can be achieved by two different means. One is you can increase the curvature of the bill itself, but also you can change the angle at which the beak is attached to the skull. And both turn out to be very important. And different beaks, different finches, actually different species, are employing slightly different strategies on how to maximize the mechanical advantage. So going back to the model, we developed this developmental model, which allows to produce differently shaped bills by playing with certain characteristics of cell proliferation inside the growth zone and outside the growth zone. And this morphospace shows both the beaks, which we managed to explain. These are of different colors, different species of Darwin's finches, which we could explain using the three-dimensional model. But in blue is the morphospace, which is available, which can be generated, these shapes, by using this particular model. This includes beaks of a lot of the actual species. We tested quite a few species outside of Darwin's finches across the birds. Some of these are very narrow, straight cones. Others are very bent beaks, or there's very deep and very flat cones. And all this can be explained by playing with different parameters of this developmental model. And we can see also there's quite a bit of white space, which is presumably not accessible using this particular model. The beak shape space, which evolutionarily is probably restricted. You probably have to change something about the mechanism of bill development to allow for those shapes. So this is what we have, and of course we are now keen to test this. We would like to look at embryos of species of very differently, of other birds which have very differently shaped bills to understand them. And another direction which we're going is, of course, to try to explain developmental mechanisms at the cell and molecular level, how this morphogenesis works. In order to do that, we need to look at the embryos. So we need to collect the eggs for this project. We've taken multiple expeditions to Galapagos Islands to collect embryos of Darwin's finches to look inside the developing beak. So this is a picture showing you the field kind of component of this, and we need to find the nests, we need to track them, we need to look at the eggs. We only collect one egg, the last one from the clutch. Then the female lays a replacement egg because she can count how many eggs she wants. So they can see finches can do some math as well. And so then we have an egg, which we then incubate in field conditions for a very precise period of time, so we can collect embryos, and the female has a full clutch, and their population is not disturbed. Some nests are easily accessible, like the one shown here, and the small cactus. Others are much more difficult, and they try to hide them inside these big cactuses. So once you have the embryo, you can then you can collect the embryos, particular stages, and you can look at gene expression. You can look at using a variety of techniques, pseudo-hybridization or immunohistochemistry. You can also do sequencing. For example, we can collect big tissues and we can subject them to whole transcriptome sequencing and bioinformatic analysis to look for transcripts which would be specific to particular species or specific to particularly big shapes. And I will show you a couple of examples of some of the genes which we previously found to be associated with big shapes. Here I'm trying to explain the difference between very deep bills that you see, what they call the ground finches. These are finches which are adapted to crack very hard seeds, and the cactus finch, which is feeding on cactus flowers. It has a very pointy bill, which it's using to kind of get in deep into the cactus flower to get the nectar and pollen. We can see that on the left are embryos from the same species. Well, this is several days before they hatch, and as you can see there, they're already making the right bills. That is, the cactus finches already have this very pointy bills as embryos, whereas the embryos of the ground finches already developing this very, very deep and very broad bills, suggesting that this bill shapes are fully under genetic control. The shape is not dependent on the usage, and in fact, when they hatch, they already have the right peaks on them. So what we found from this work, and I'm just will show you two different genes from now the list is quite a bit longer, but these are two kind of interesting key stories. Again, we're comparing, looking at 4 species. So here's the ancestral species. We know that Darwin's finches are originally from Central and South America, and they're related to this little grass quits. These are very small sparrow-looking birds which feed on grass seeds on the continent, and they evolved into the sharp-billed finch, which has a small symmetrical bill, or the large ground finch, which has the super deep bills and very broad bills, and the cactus finches, which have elongated bills. And of course, the two gene stories. One is BMP4, the gene which is important for controlling skeletal structures. The BMP4 means bone morphogenic protein 4. And we found that this particular gene is expressed, that is activated much stronger and much earlier in the large ground finches, in those deep bill finches, but not in the more ancestral form, and not in the long bill form. But in contrast, another gene called calmodeling was upregulated at very strong levels in the species which had elongated bills, as shown here with a little red arrow. So both of these changes are novel, because this more ancestral species of finches which don't have very deep or very long bills, don't have this very strong expression of these particular genes. But the question then was, are these molecular changes, these differences in gene expression, are important? To address this question, we wanted to mimic this mutational changes in our laboratory model system, the chicken embryo. So the chicken allows us to, you can grow the embryo and allows us to manipulate with development. That is, we can, for example, use a retrovirus which carries a gene like BMP4. We can infect the tissue inside the embryo, and we can upregulate or downregulate a particular gene in the developing phase. So what you can see here is the normal chicken embryo at late stages, looking from the side and looking from the front. The chickens normally have a fairly narrow bill, which is adapted to pick up small seeds from the ground. Now, if we upregulate BMP4, and to remind you, this is the gene which is upregulated early and strong in the ground finches, in those bullfinchy species. What happens if we do exactly this? If we upregulate BMP4 and make its expression stronger and earlier in the chicken embryos, if you do that, then they will develop a finch bill. This chicken embryo here has a much deeper bill and much wider bill than the normal chicken embryo. And if you do the opposite, if you downregulate BMP4, you get the opposite. You actually get a bill which is narrower and less deep than normal. In fact, but because we can control exactly how much infection, that is, how much we can gradually dial up BMP4 by controlling the amount of infection the embryo sees in the beak, we can go all the way from normal looking bill to deeper bill, which seems like a kind of a small finchy beak, all the way to this very, very bullfinchy beak, where you have this very, very deep, very curved bill that looks like a finch beak on the chicken embryo. So this suggests that, in fact, BMP4 upregulation is quite sufficient to explain that particular morphological change that is highly adaptive in nature. And we did the same thing for other genes. For example, co-modeling, I mentioned a gene which is upregulated in longer billed species. If we upregulate co-modeling, you get a longer bill. And since then, we found that these molecules like BMP4 and co-modeling, they control earlier aspects of bill development. They actually control the cartilage, which forms before the bone. At later stages, the bone, the big bone, is regulated by a distinct set of molecules, which we also tested. And combined together, they help us to explain how this three-dimensional beak shape is achieved, how these different dimensions are regulated. So this is again, pictures suggesting that the beak development is complex. So from late to mid to late stages, we're seeing cartilage here, shaded gray, playing a role early on. Then the bone forms this brown, in different species. It's different dynamics. In very deep bills, bone is prevalent, whereas in the longer bill species, cartilage is the main tissue. And of course, all these genes like BMP4 here, beta-catenin, co-modeling, they all play a role in regulating distinct beak shapes in different species. And we can explain, because we know if we can co-modeling, we know how much of the beak length it will increase from experimental studies. We can then compare it with the actual increase in length of the bill in Darwin's finches, and we can say, well, we can actually explain that elongation by because basically action co-modeling explains most of it, so we can match this. So what we've been doing more recently, we were using RNA-seq to look at just focusing on the developing bills, trying to look for additional new genes, new transcripts, which are specific to different beak shapes. So these groups are referring to those groups of shapes which can be collapsed into each other by scaling alone. So these are the scaling groups, multiple species, and we indeed find interesting transcripts which are specific to particular beak shapes. So what we're trying to build here, what we're trying to establish, is a network of genes which control beak shape development in Darwin's finches. So we're finding that there are indeed multiple genes involved. Some of the genes are the ones which we found early on, like BMP4 here, beta-catenin here, co-modeling. These are some of the newer genes. The little red stars suggest some of the important genes which are key for that allow interaction between these different molecules. So this this modular network is growing, and we're hoping eventually to understand how the genetics essentially behind different beak shapes using this approach. Another dimension which we're taking out right now is trying to explain other birds. We started with Darwin's finches. It looks like we can explain them relatively well, but what about other interesting species? So here is another example of bird evolution going crazy. This is happening in Hawaii. Hawaii they were about 10 million years ago. They were colonized by a finch from Asia, and it then evolved into this many species with very different beaks. Some of them are generalists. They feed on different types of diets. Others feed on finches. In fact, this was primitively a finch. And the most basal species here are finches. Others, they evolved as curvy bills to strip bark from the trees and look for insects underneath. These are creepers. And eventually from these creepers, independently several times, these honey creepers evolved, which evolved even longer. This very beautiful sickle-shaped bills, which they used to insert into these tropical flowers and drink nectar. And they became nectar-feeding birds. So this is a phylogeny shown here on the left. Again, this entire Hawaiian clade evolved from a rose finch-like bird from Asia. The basal groups are still finches, like this Laysan finch. Here's the skull of the Laysan finch. And then many of them became creepers. And then independently, some of them became honey creepers, which have these very beautiful beaks. The challenge here is that it's not just the bill which is curved. We can explain the Finch bills by using our original model, but how do you explain the fact that the entire bill essentially curves? In fact, if you look at Darcy Thompson's book, he has he refers to this particular type of transformation, geometric transformation, as radial bending, where the entire structure, it's not just scaled, it's not just bent, it's actually it's not just sheared, it actually bends around almost like a circular surface, and that's something that we are hoping to now. We're just beginning to look at this species. We just recently completed scanning Hawaiian honeycreeper species. We're hoping to also eventually get to their embryos to try to explain this group of birds. And that's basically where we're heading as well. So we're hoping to look at other additional examples of bird diversity, looking at their beak shapes. So just to summarize the logic behind this types of changes, what we found is somewhat surprising and perplexing, because we found that as a field, that structural genes, the genes which control bone development and cartilage development, muscle development, are essentially the same across all animals, certainly across all vertebrates. We also found that regulatory genes like BMP4 and calmodeling, other regulatory developmental genes, they're also highly shared. They're essentially the same genes. We have BMP4 and calmodeling, and so do all the other birds. And yet they produce, when you combine them, this highly conserved sets of genes, they produce all the diversity. How is it possible? Well, the best analogy I could find is architecture, construction, where we use the same building materials. We use wood and cement and bricks. We use the same tools even, and yet we produce very different looking buildings. And of course, the main difference is in the blueprint. So there is a plan and when, how, and where to use the tools, which makes all the difference. And it's all, so in biology, it's all about these regulatory programs, these genetic blueprints that allow to use regulatory genes, the same regulatory genes, very differently in different species, which eventually produce this beautiful adaptive differences, which you see during evolution. So I'm now going to move to yet another story, just to illustrate more to this point, how yet another group of diversifying animals, in this case bats, can be explained by both morphological and also developmental level. So this particular project is on a group of bats called Phylogeny. These are leaf-nosed bats from South and Central America, and they have this absolutely crazy faces. You can see here, many of them look like aliens. And if you look deeper, if you look at the skulls, it turns out that the skulls very carefully reflect the different adaptive features of the face. So primitively, this group evolved from a bat which feeds on insects. As you know, bats use echolocation. They hunt at night. They use echolocation to locate a small insect in the air, and they snap it, and that's how most bats feed. And this is exactly how this particular group, Phylogeny, started. And we still find bats at the base of this group, the most primitive of the phylostomates, which still look like, they have this really interesting leaf nose, and they have an unusual way of echolocating because they release sound from their nose rather than from their mouth. That's why they have this, beautiful leaf nose. But other than that, the diet is normal for bats. From this insect-feeding bats, they evolved into bats which have super long snouts. As you can see here on the left, these are the bats which insert the entire snout and face into the flower to drink nectar. Others have very short faces, essentially flat faces, very primate-looking skull, and they feed on large fruit. Here is a bat which is several actual species-related bats or vampire bats, which feed on blood. Their skull is very unusual as well. The upper face is bent, so the incisors, the front teeth, are pointed forward. And they're very sharp, so it allows this bat to drill a hole in the skin and drink blood. And finally, this is the flying wolf of these bats. It's the largest bat from this family. It has a carnivorous skull, complete with this big canines and shearing teeth, and it hunts rodents such as rats and hamsters. And it's a real carnivore. So you have this amazing diversity of faces in one family. And one reason why this is exciting is because this appears to mirror what happened early in the mammalian evolution. Back, we're talking about over 100 million years ago, when this different orders such as carnivora, right, which includes cats and dogs, or Cetrodactyla, this is the whales and relatives, or this other primates, this very different looking orders of modern mammals evolved from. Nowadays, if you compare a monkey with a wolf and with a whale, they're completely different. They have very different skulls, very highly specialized skulls. You'll be comparing apples and oranges. And yet this group of bats here, they evolved skulls which are carnivorous, or these elongated skulls, very whale-looking skulls, or the skulls which look like, the skulls of this insect-eating or primate skulls. And yet it's all within the same family. It's all within 10, 15 million years perhaps. So by looking at these skulls, we can better appreciate and understand what happened very early in evolution of mammals themselves as a group. And of course, we would also like to explain this adaptive radiation, this adaptive diversity in this groups of bats. So here's a phylogeny, and it shows that all of them started with as insect feeders, this kind of light orange, and then they evolved. Some of them evolved to maybe vampires or they're carnivorous bats. The nectar feeding with super long faces bats evolved at least twice within this clade. A lot of interesting evolution going on. To explain it, we also generated a full three-dimensional CT scan library of all the species, not all species, but many species from this clade. And then we put, in this particular analysis, we placed, because it's so rich with landmarks, we placed well over 100 landmarks around the skull, both outside, inside, around the brain, around the nasal structures, because the noses change. Some of the bats, instead of relying on the echolocation to find insects, they begin to rely more on noses to find those flowers or fruit at night. So there's different parts of the skull and the brain and ears change in different directions. So we'd like to kind of map this. So we put lots of landmarks around the skull. And here's the morphospace of 3D shapes. And you can see here that the beginning point, the little red pentagon, is the original position of all this diversity, right in the middle of this morphospace, and from which point it literally radiated in all directions. Some of these became very short-faced and became food eaters. Others became very long-faced and started feeding on nectar. Here's a vampire bat here, Desmodus, on the left, very unusual face. This bat here in the bottom right corner is one of this carnivorous wolf-looking bats, which feeds on mammals. Here's a bat which also feeds on prey, but it feeds on frogs and soft-bodied prey, and has a very different looking skull. So this is a lot of interesting evolution happening here that we're hoping to explain. And if we look at the principal components, that is the main dimensions of change. The PC1, this one, the x-axis, is primarily about the length of the face and the snout. So the skull in the middle of that space looks like a normal bat, whereas in the extremes it looks either a bat which has essentially no snout, completely flat-faced bat, like some of these advanced fruit-eating bats, or this super long, elongated, super long face bat that feeds on the nectar. So this is all kind of along this, it's all about the length. If you look at PC2, the y-axis, which also explains a fair bit of variation, it's all about how the face is oriented relative to the skull. So some faces upturned, like in the vampire bats. In the middle is the ancestral orientation. Some of them are faced downward or upturned. And this should remind you, remember about how the beaks were connected to the skull in the Darwin's finches. So that dimension is also very important in these bats also. So next we wanted, as development biologists, to try to explain this from the cellular and molecular perspective. So what we did, we collected embryos of different species, several different species of allostomid bats, which represented different shapes. And what we found is an example of what's known as, see if it's, yeah, sorry, I skipped this slide. So what we collected a number of embryos from different stages for each species, and we looked at embryonic skulls and juvenile skulls. We put the same landmarks on the skulls as in the adults to understand how the skulls essentially acquire their shape during development. And what we found is an example of what's known as heterochrony. Now, heterochrony is a phenomenon whereby changing something about the timing of development, when you speed up or slow down development, or you change exactly when a particular developmental process begins or ends, we can produce interesting evolutionary results. For example, acceleration here, so here in yellow is the ancestral developmental rate at which the phase develops in this bat which feeds on insects. Whereas in blue, you see the bats which have very, very, for example, these very short phases, which have a certain stage of development, they have much faster growing phases. And that's something called acceleration. And it's an example of heterochrony. Whereas bats which, this very long phase bats, such as the flower-eating bats, nectar-feeding bats, so if you compare the yellow ancestral trajectory versus the purple one, what you see is that the development becomes extended. So they continue to grow their faces for significantly longer. It's something that's known as hypermorphosis, and it is also an example of heterochrony. It's also an example of changing timing of development, which produces either a very elongated face, or if you continue to bend the face upward, it will produce a face of a vampire bat, which also rely on hypermorphosis to produce their unusual faces. So these are textbook examples, essentially, of heterochrony. The hypermorphosis, when you extend development. For example, when the bats which feed on nectar, when they're born, their face is too short to feed on flowers. They have to stay in the cave and wait for the mother to feed them for several months before the face becomes long enough to actually feed on the nectar. So that they have extended the developmental trajectory to produce their novel faces. Whereas short-faced bats, they accelerate through development. They develop faces slightly faster, but they end up with shorter faces. And to understand how that's possible, we wanted to look at one level below. That is, we wanted to look at the cells and tissues. So if you look at the skull, these are the structures inside the skull which support the face. These are the nasal septum, some of the other cartilage and bones, which are indicated here. This begin the development in the embryos. This is the embryo of a bat, a nasal cartilage here, cartilages of the cranial base. So we wanted to focus on the cells inside the embryo to understand how this morphogenesis, the formation of the shape, was taking place. For this, we wanted to collect embryos. This was done by now my former PhD student, Jasmine Camacho. She went to different Caribbean locations, and you can find bats in these old water tanks. There are colonies of hundreds and hundreds of bats living inside, obviously in caves. When you have multiple species of multiple members of multiple species living side by side, and they established the lab spaces and on campus of University of West Indies, where they would collect embryos of different stages and preserve them for laboratory analysis. And so back in the lab, an embryo could be taken. We could section it into, the entire embryo could be sectioned into slides, and then we could image proliferating cells and then stack them together to produce a three-dimensional view of cell proliferation inside the developing phase. And we wanted to compare the embryos from at least three different species, the one which has a very long phase, the one which has a normal face and a skull, and the one which has essentially very, very short face. So this is the skulls of the adults of the species that we subjected to this cellular analysis. And we also had an outgroup, a normal bat related to the Trophilostomys. So these are heat maps which show proliferation, how it changes from stage to stage. You can see it's a very dynamic proliferation. There are these organized cell growth zones. Early in development, you can see a lot of proliferation in the brain. This is the phase kind of looking to the right. This is the future phase. That's the brain here. There's a lot of proliferation early in the brain. Then it begins, then you see a lot of proliferation in the face, and there's again dynamic changes of what happens to this growth zone inside the face. But we know that it is these stages which are critical, because at some point cell proliferation, essentially organized cell proliferation, stops in the face, and they just begin to expand, and again in a way that's similar to what we've seen in birds. So we focused on different cell populations inside the developing head, look at the brain versus the face versus the back of the face. So we wanted to understand the kind of full dynamics of cell proliferation in each part of the head. And we found, this is just again looking just at the face here. That's the area in which we're calculating cell proliferation across multiple stages in different species. And what we found is that compared to cell proliferation in the more ancestral species, what we found in some species, cell proliferation was accelerated or decelerated. And on the next slide, I'm going to try to make sense of this, but we were able to detect distinct differences in how much cell proliferation was detected in the developing phase. So here's the model. This is how we managed to explain this. So in the phase that is, this is the short phase, the relatively short phase of an ancestral species. The cells go through 3 different stages. In green, these are the early cells which are proliferating, and they're completely naive. These are these are progenitor cells. Then the cells become skeletogenic. They commit themselves to produce skeletal cells, but they're still fairly naive. They're still proliferating. And then finally, in the red are the cells when they begin to form bone and cartilage, and they stop proliferating. They just form kind of a lot of matrix, and they make bone. So what we found is that in bats which have even shorter faces, those are the bats of the fruit-eating bats, faces of fruit-eating bats. What we found is that they go through this proliferation steps, especially when they enter the skeletogenic stage, when they are in this orange part, and they accelerate through them, and they finish it much. They go through the stage much sooner, much faster, and they enter the next stage when they begin to lay down bone much sooner. So they end up with a shorter but more bony face. So that's the long of these super long faces of nectar-feeding bats. What we found is that the early stages were normal, untouched, but it's the stage at which the future bone cells were still proliferating was extended. Indeed, it was continued all the way through embryogenesis and probably ended some way well after they were born. So this is, again, so what we're seeing here, we're seeing really nice examples of heterochrony at the developmental level, which helps us explain heterochrony, which we detected at an evolutionary level by comparing multiple species of bats. To move even further, we wanted to understand which genes may be involved, and we looked at multiple signaling pathways which control cranial development. We found that BMP pathway appears to be, so we found evidence that BMP pathway is activated differently in different species of bats. I'm not going to get into tons of detail. We just found that in a more basal species, this is our baseline. We found that in species which have very long faces, it was upregulated BMP pathway, whereas in the bats which have very short faces, it was downregulated this pathway. So we have very distinct levels of BMP signaling in bats with different faces of different lengths. So that led us to the model where we have kind of very early events are very similar. So remember those neurocrest cells which come to the face, they begin to proliferate. The face early on looks the same in all the species, but later when they begin to proliferate and morphogenesis begins, when they begin to form those growth zones, then the behavior changes. So in some species, it stays kind of ancestral levels. In other species, proliferation is sped up and finishes earlier. In other species, it's extended for much longer periods of time, and it appears that this shift in cell proliferation is regulated by BMP pathway. To test this hypothesis, we are now generating BMP mutants. So in mice, we can play with this pathway genetically. So if we upregulate BMP in the face, we begin to see faces which are now longer than the wild types. When we downregulate BMP pathway, we begin to see faces which are shorter and shorter. So because there are actually 2 genes which control the face, BMP2 and BMP4, we need to play with, try to suppress more and more alleles of these 2 genes, but we have definitely seen shortening of the face when we downregulate this pathway. So in order to quantitate this, we put landmarks on the developing embryos of mice, and then we're seeing how BMP, looking at mutants versus wild type, are helping us to. So, for example, when we produce a shorter-faced mouse embryo, mouse mutant, we now can compare it more meaningfully using the same landmarks as the faces of short-faced bats. So that's where we're kind of heading with all this. This is very much in progress. But the point is that we're trying to see if we can explain that 3-dimensional variation in facial shapes in these bats by looking at the mouse model where we play with this very pathway. which you think is involved in evolution of the face. So to summarize, this is a developmental model for facial evolution and diversification across all amniotes, all the land animals. What we're seeing is that early events are very conservative, right? But it's the changes of the very, very early steps, where neural crest cells go and how do you begin to form the face that explain the difference between different classes of amniotes, such as birds versus crocodiles versus mammals. Later on, once you kind of lay down that foundation, the exact proliferation within the development phase is critical to establish this adaptive variation that you see in the adults, faces which are longer or shorter, faces which are wider or narrower. All of those differences can be explained by changes in cellular proliferation and cellular differentiation, which are happening around mid stages of development. So the one of this way we're doing, pursuing this project, we're now beginning to create a map of cell proliferation across all the different amniotes. So I showed you results from birds. We would like to add more birds. Of course, I showed you results from mammals, such as bats. Would like to look at more mammals. I haven't really talked about, and we have a very lively project on crocodilians. So we have access to multiple species of crocodile embryos, where we can look at the proliferation, cellular proliferation phases of crocodiles with very short faces or very long faces, like those garter-like species. If you're curious about this, I can tell you more. But now I'd like to step back and see if we can, what are the kind of major conclusions we can draw from this kind of studies. So first, biological shapes analysis useful for evolutionary studies range from simple linear measurements to landmark-based and landmark-less approaches, which allow trade-offs between phylogenetic scope and 3D precision. So again, how you capture the shape, right? So we early on we were looking at landmarks. We were measuring literally how long or short the face is, looking at two-dimensional outlines. Now we're moving into, thanks to those CT scans, we're moving into kind of three-dimensional methods. It is important, indeed critical, to include ontogenetic data, that is developmental data, to make sense of the evolutionary variation, no matter what phylogenetic scale. So this is how we, for example, discover those heterochronies, by comparing both evolutionary trends, but also by including juveniles and embryos. That when we noticed that in some species, the development was slowed down or sped up. Analyzing morphological transitions within both evolutionary and developmental timescales reveals widespread deployment of modular heterochrony. So again, we now looked at birds. We looked at mammals such as those bats. We looked at crocodilians, and we're finding that playing with heterochrony, playing with exact timing of developmental events, apparently is quite widespread and very, very important for generating adaptive variation in those animals. And finally, uncovering cell and molecular level mechanisms for morphogenesis helps to better understand such phenomena such as modularity, heterochrony, and evolvability. So in our studies, we'd like to combine different types of approaches. So in orange are the geometric morphometric approaches, again, which range from linear measurements to three-dimensional morphometrics. These are then matched by this green methods shown in green, where we compare embryos. Actually, this is shown in orange. So the blue are morphometric methods. The orange are the methods of comparative embryology, where we compare embryos, developmental strategies of species, different species, to understand which developmental changes can help us explain those morphological variation. And finally, in green are the experimental methods, where we take those candidate genes, take those candidate cellular processes, and we subject them to experimental analysis. And we try to essentially explain those morphological changes which we see at that level. And then, of course, it keeps on going. The more we learn about any particular process, the more questions we can ask, and so forth. And again, this is some of the projects which, some of the different types of animals we study. I told you about the finches, a little bit about the honeycreepers. I told you a little bit about the crocs, the bats, but we also have projects now looking at lizards, crocodilians, and the eventual goal is to try to explain the faces from the early point until late point, and from variation, trying to explain how why birds have very different faces from the mammals or reptiles, and also why closely related species such as Darwin's finches have very different faces. That's it. So I'd like to acknowledge members of my group, in particular, Jasmine Camacho, who was working on bats. Ashley Rennie was a PhD student working on birds. Maria Dobra was a postdoc, also working on birds and Darwin's finches with me. Of course, our field assistants. Also, many of our collaborators, in particular, postdoc with Dr. Mahadivan Salem, who did a lot of modeling and biomechanics work for the Darwin's finch project. So I'd like to thank our funding sources and, of course, you for your attention.
Great. Thank you so much, Arkhat. That was an absolutely amazing talk. Any questions from the audience?
Yeah, I'd be happy to entertain any questions.
I wanted to ask a quick question.
Absolutely.
Can you hear me okay? So I was wondering about bills. The finches you looked at have more or less all the same shape, but some birds have kind of strange shaped beaks, like I think they're called the spoon bills. And so had you thought about like how to modify the model to encompass something like that? Because you get that non-linearity at the end of the beak.
Yes, so you're right. Our model explains conical shaped bills. So the paradigm here, the program which we first found in zebra finches explains conical shaped bill. So it involves this very large growth zone, it decays into nothing, and it creates a little cone. Other types of bills, such as those spoon bills, right? The bills which no longer look like cones, obviously will require a complete repatterning of the system. How they do it, we don't know, and I'll be really, curious to understand them. My favorite example, like a challenge I would like to at some point address, is like a flamingo bill, right? It's a completely, this unique adaptation, right? It's using its bill upside down necessarily to, scoon out, plankton from water, and it's shaped in a way that's not complete. So there are interesting shapes which we cannot explain for sure. Right now, we're just trying to see how well we can explain conical shaped bills, but I suspect to allow for those unusually shaped bills, very different shaped bills, you'll probably need to rewire the entire, it will need to rely on a different cellular mechanism. How it works, we don't know.
So Betsy Shock has a question. Have you looked at species-specific differences in enhancers for genes such as BMP4 that could help explain differences in signal duration or localization?
Yeah, great question. So going into kind of further molecular mechanics of all this, so we have a collaboration with Leif Anderson, who, in fact, there are a couple of papers or joint papers coming out literally this month with Darwin's finches. So he is an expert in comparative genomics in Darwin's finches, and we looked at, so at the moment, they're primarily looking at the genes which are under selection, which also, whose variation, genetic variation correlates with morphological variation, let's say in big shapes or pigmentation. They have multiple projects looking at different aspects of finch evolution. So the one thing which we are finding is that the molecules which we know as sort of directly involved morphogenesis, molecules like BMP4 and co-modeling, are probably not the ones which are directly mutated. That is, mutational loci which we picked out so far, and we are still testing some of these kind of functionally, are interesting genes, but not necessarily the genes which we know are proximally involved in morphogenesis. So it could be that the mutations which ultimately cause upregulation of BMP4 are actually 1 or 2 steps removed genetically from BMP4 itself. So that's something that, and I told you about the network, right? There's so many genes. There's actually, these genes do not work in isolation. None of these genes do. So each of these genes regulates multiple genes, and these networks, they interact with each other. So I think it's not going to be simple as finding a mutation that controls BMP4, unfortunately. We looked for it. We looked at BMP4 locus, one of the first things, obviously. But what's emerging is that it is this network which changes its behavior, and there will be kind of mutations which will affect the behavior of the entire network. There's similar stories emerging, for example, pigmentation, right? Genes like MC1R, which controls like 200 plus pigmentation genes. Exactly how mutation MC1R trickles down to a particular pigmentation phenotype is actually sometimes complex. We're probably facing the same thing, which will be a network level change. So again, there is a set of genomically identified kind of genes which we're testing right now, which we're hoping will be part of that network which regulates molecules like BMP4, which will eventually be the answer. Hopefully that will, that answers your question.
She says thank you. I have a quick question. Well, I don't know how quick it is. I was wondering how bits of the brain case and growth of the brain case could impact the growth or the shapes of the facial features, especially in the case of where you were talking about, the biomechanical consequences of the angle of the beak mounted on the brain case. And situations where you have species like in the bats that are relying on different kinds of sensory information, and so you can imagine different parts of the brain are expanding to process that information, which could be pushing on the skull and affecting the brain case shape. How have you looked at how those different modules interact to impact?
Great question. We know they do. We know that the brain interacts with the skull and vice versa. So these two domains necessarily talk to each other. They have to fit this kind of famously as hand and glove.
Exactly.
But this relationship is not an easy one. So we know about this relationship, for example, from biomedical literature. So famously, psychiatrists in the past were told to look at the face to recognize a lot of the otherwise psychiatric psychological syndromes, because because the craniofacial development often parallels what's happening in the brain, especially the facial part. So I haven't shown those slides where, for example, in bats, because we have landmarks around different parts of the inside parts of the skull, the different parts of the brain change dramatically sometimes, because there's part in front of the full brain, which is used for olfaction, for sensing smell, and that part is very small in the early bats and expands like this giant bulbous looking structure in bats, which rely now, like those nectar feeding bats, they have to find a single flower in the middle of the night, right? Sometimes like several miles away. Or those vampire bats, they actually find their prey first by smell, like large mammals, and then they can switch on other senses. So different parts of the brain expand or develop to different extent. So this is something that my now former PhD student, who is now very actively doing the postdoc, Jasmine, she's taking that direction. So she's very much involved in I'm part of that collaboration. So, but it will be one of the things that she's very interested in. In fact, looking at things like nasal epithelium, like nasal structures, right? How they switch from what happens with the middle ear structures when they stop no longer rely on precise echolocation, but instead switch to other senses to find food. So all these things are very much work in progress, but absolutely. So I think there's different parts of the brain and how they how they affect directly or indirectly affect the face is very important. Vice versa, if you famously biomechanically, if you switch to very heavy master muscles in mammals, like this very heavy chewing muscles, the way how they work is that you have they attach here to the jaws, but they fan out, they connect here, and famously in carnivores, there's this very strong bony crest which forms on top of the skull that that anchors this very powerful master muscles. Now, because they form this crest, they have to fuse the skull bones early, and those and that actually places a lot of constriction on the brain itself and how much it can expand and things like that. So there's a reason why we don't have the such crest, and our jaw muscles are very, very weak, allowing us to actually but then we rely on processed food. And we don't have muscles strong enough to chew like a potato or a piece of raw meat, because our muscles are puny by comparison, and but that allows us to keep the sutures open for much longer, and allows us to expand our brains for much longer. So there's you can see that it's a really interesting but rather complicated relationship. So yeah, absolutely, it's something that will require some additional investigation. Yeah,
absolutely. Thank you. Well, I think we've lost quite a few people because they have to run on to other things.
that's fine. Yeah,
but thank you so much for an absolutely amazing talk. Those visuals were spectacular, and that experimentally and mathematically rigorous. research. So it was very enjoyable. Thank you so much for your time.
You're welcome. It was a pleasure. It's, as I said, this is some of the most exciting projects, because we get to talk to people who use very different skills to look at the same problems, and it's exciting.
Absolutely.